Thank you for attending this virtual poster session. My name is Vibha Anand. I'm from IBM Research. This work was done in collaboration with our partners at the Cleveland Clinic. And I'm here to talk about LEOPARD, which stands for Linking Experimental Observations with Predictive Algorithms to Reveal Response Dynamics for the Single Cell. This work is motivated by advancements in nuomics technologies that have evolved along with AI methods, resulting in biomedical foundation models, or BMFM. BMFM learn universal representation of omics data, and via transfer learning methods provide tremendous opportunities for accelerating scientific discovery. Indeed, one such model called BMFM RNA has been recently released by the research group and forms the computational core for this work. It is presented here as a repeatable framework. I would like you to focus on figure 1. At the top of this figure are data and models of disease. We need to start with a BMFM pre-trained model that can be adapted or retrained and fine-tuned to the specific needs. LEOPARD, in fact, is an evolving biomedical agentic framework for the single cell analysis. It is designed to leapfrog roadblocks in making discoveries by exclusively using human data and employs the foundational capabilities of BMFM RNA, such as to transfer biological labels of cell type or health status from human samples in vivo studies to in vitro cultures. In future, it can iteratively also perform gene regulatory network studies and enable gene perturbations. As in figure 1, 3 methodical agents are derived from BMFM RNA, and they are used to demonstrate our early results or translating human in vitro and in vivo studies. The first agent, called Discoverer, identifies key genes that are important in terms of rank order for the cell type under development or in vivo cell. The predictor agent quantifies the expression of the gene due to in silico perturbations. In this work, we knock out a key transcription factor for cell development in the in vitro study. And the comparator agent, which is the most developed agent of all three, quantifies proportionality and similarity of cell types between datasets at hand. In our use case, the reference dataset is derived from in vivo study and the query dataset is derived from in vitro study. I would like to talk about the procedural steps outlined in figure 2 and the metrics that we used to measure the similarity and proportionality of these cell types. But before that, I would like to focus on the study design and models on the right panel. So we have previously used studies, single cell RNA-seq studies. In this work, these studies were conducted using biopsy samples from human epithelial colonic cells derived from healthy donors, that is the in vitro study, and healthy and diseased donors from the in vivo study. Our results focus on a specific cell types called the goblet cells. Translational validity of this work is explored via model data splits for the fine-tuned model performance, also listed in table 1. Now I would like you to focus on figure 2. So in this work presented on this poster, figure 2a depicts the in vivo study in which the cell types were manually cell typed. On the panel figure 2b, we have a reference data set which is an in vivo study of the human epithelial colonic cells. And in figure 2c, we use the foundation model BMFM RNA using model data splits to fine-tune the model and use it as a classifier to classify the cells in the in vitro study and vice versa. In figure 2D, we show our metrics for comparison or understanding the concordance between the in vitro and the in vivo study, which is done using three different metrics. The total labeling concordance, which measures the entropy in the clusters and label in all samples and mutual information between labels transferred and the cell type clusters. The cell type labeling efficiency, which measures average probability of a cell type label from a reference dataset given there is a cell type cluster in the query dataset. The conformance, labeling conformance, which measures a predefined confidence level such as 95% and gives a conformal set of cell type labels per cell sample. In this poster, we do not talk about labeling conformance, but we present results from labeling concordance and cell type labeling efficiency. Coming back to our results, we had 5 fine-tuned models where the average modeling performance is listed in table 1 for both the in vitro model and the in vivo model. The overall entropy in cluster and cell type labeling is shown in table 2 and is significantly different between the two in vitro datasets we obtained, which is day 7 and day 2. The mutual information measure, which measures how labels align with the clusters, shows an increase between day 2 and day 7. This indicates that in vivo labels, when transferred to the query dataset population of cells, show stricter correspondence to the intrinsic differences that are found in vitro data clusters. In other words, as the cell differentiates between day 2 and day 7, we are finding that the labeling of the clusters and the labeling derived from our foundation models are very coherent. This information is depicted in table 2. To further understand the labeling efficiency at the cell type, please focus on figure 3. Here we describe the labeling efficiency of goblet cells. So we have 5 models that could transfer the labels from in vivo dataset to the in vitro dataset and vice versa. And these 5 models show high labeling efficiency for most cell types, but only goblet cells are depicted on this poster. The average cell type labeling efficiency for goblet cells across 5 models is also very high. It is in the order of 0.93 for in vitro to biopsy and 0.90 for biopsy to in vitro, thus highlighting a high correspondence between the goblet cells found in vitro cultures and in vivo samples. Through the other 2 agents, which are the discoverer and predictor agents, we also found the important genes that may be involved in goblet cell development at day 2. Of them, 1 gene was also found in an independent analysis differential expression of gene at day 7, in day 7 data. The knockout of an important transcription factor in the day 7 data set in vitro data also label most sample cells as progenitor or stem cells in our predictor agent. So these results indicate LEOPARD as a viable framework for conducting experiments to compare in vitro and in vitro datasets. Cell type annotation is a critical task and is often performed manually. Manual annotation is time-consuming and laborious process and often susceptible to the choice of clustering method, parameters, expert subjectivity, and the use of non-standardized cell type ontologies. These can introduce bias and make results non-comparable across datasets and experiments. Using the LEOPARD framework, we develop a disciplined approach to mitigate these biases and accelerate the scientific discovery. In this preliminary work presented on this poster, we have shown that how in vitro and in vivo samples can be compared across cell types. Experimental design using LEOPARD framework also opens doors for many other biologically inspired investigations, and therefore addresses an unmet need, that is to compare fidelity of these in vitro systems with human biopsied in vivo samples. Needless to say, such comparisons can also inform decisions to optimize expensive in vitro investigations for making future discoveries in many chronic diseases. We believe LEOPARD framework can open door for many biologically inspired investigations in the future. Thank you.